From February 13th to the 15th, EHC staff and members of the EHC Steering Committee entered the fascinating world of Genethon, a laboratory in Évry, France, dedicated to advancing gene therapy treatments for rare diseases. Since its creation in 1990, work done at Genethon has greatly contributed to understanding and decoding human genes, the regions of our DNA that contain the information needed to make proteins or other molecules essential for the growth and functioning of an organism. The 200-plus researchers, pharmacists, doctors, bioproduction engineers, and technicians that work there continuously build on academic discoveries to develop them into gene therapy treatments for patients. There are currently 6,000 to 8,000 known rare diseases, 80% of which are genetic in origin. As gene therapy holds many of the answers that the hemophilia community has long searched for and could even be a potential cure, we in turn had many questions on how it works and what its challenges and successes look like. We signed up for training at Genethon with the aim to understand more about this exciting novelty and to be able to better break down the concept to patients and our national member organizations. But before getting to the main focus, we were first introduced in depth to cells, chromosomes, sequencing, amino acids, proteins, and more head-spinning human components that are so complex they make you want to pull your DNA out. It all starts with cells, which are the basic unit that makes up all living things. Each cell contains the same genetic information, located in the cell nucleus, in the form of small structures called chromosomes. Humans have 23 pairs of chromosomes that we inherit from our parents and that determine our sex, eye and skin color, susceptibility to diseases, and many other genetic traits that make us who we are. The chromosomes themselves are composed of threads of DNA whose language is decoded by mapping out the sequencing of 4 chemical substances called nucleotides or bases: adenine, thymine, cytosine, and guanine. In each of our cells, our DNA is composed of about 3 billion of these base pairs. Without getting ahead of ourselves, it is a mutation, which we learn can happen in several ways, in the order of the base pairs that causes genetic diseases. To help us visualize these concepts better, our trainers at Genethon had us do a series of experiments. Our first task was to isolate our own DNA from our saliva, which is surprisingly easy and can be done even at home by adding dishwashing liquid, a pinch of salt, and rubbing alcohol. Another experiment was isolating a specific area of our DNA and amplifying it in order to analyze the number of base pairs located there. In the general population, this particular DNA fragment either contains 100 base pairs or 400. The experiment showed that we were a very homogeneous group, as all of us had the short version of the fragment with 100 base pairs. Claire Rochette, a scientist at Genethon, speaks in more detail of the experiments we did in the 3 days and their lead-up to learning about gene therapy. Did a lot and learned a lot for these 3 days. Yes. Before we even got to the topic of gene therapy, there was a lot of other concepts we had to cover first. Yes. Can you summarize just briefly, I know it's hard, in a minute or two, of what we did here? We started with learning about cells and chromosomes and DNA.
Exactly. And to know about DNA, you made your own extraction of DNA from your own cells from mouth. Mm-hmm. And you can see your DNA as a jellyfish in a tube. And so you will remember this experiment because you have done it. Yeah. It was very easy.
It's very cool.
Okay.
Yeah.
Yes. And the day after, you analyze your own DNA with an electrophoresis system. And before you amplify your DNA, a special part of a chromosome 8 of your DNA to see if there is a mutation on it. So you have done it yourself and it works. And in this afternoon, you have put a new gene, GFP gene, in bacteria to do the link between DNA and protein. To do protein, to make a protein, you need a gene, special gene, and you can put a gene in bacteria, which is a cell, to produce protein. And today,
holds the information.
Exactly. And this new information changes the phenotype of the bacteria. And this morning, you have seen the result of the experiment. You have seen bacteria colony, a green colony for the positive control, and you have compared with the negative control with white bacteria on the petri dish. So now you are sure that it's the gene you put it in the bacteria that changed the phenotype of the bacteria.
So we need to know the role, obviously, of the gene.
Exactly, to know the role of the gene, and it's the same way to produce a factor for coagulation by Baxter or some big pharma. They put DNA coding from factor 8, for example, for coagulation in bacteria or yeast or hamster cells, and they produce human factor for coagulation as you did with the GFP protein.
It sounds so simple, but not in practice. Gene therapy in hemophilia is a relatively new concept.
Yes, exactly.
The community, but it actually goes farther back. Can you, when was, or how did gene therapy come to be?
In France, the first taste in man with gene therapy was in Necker Hospital with Alain Fischer, the clinical researcher, and he treated 9 children with a special rare disease of the blood cells, white cells, called SCID-ADA. It was in 200.
This is cancer.
It's a sort of cancer, yes. And now for hemophilia, Elena Bardon explained us that the first trials in man was started in 210 for one form of hemophilia, so 10 years later.
Okay. And in a very simple terms, since we're talking about this, can you say what gene therapy is? Just in very brief.
So gene therapy is the way to put in nucleus of patients in every cells you need to treat DNA without any mutation. Okay, so it's DNA. But if you put DNA on your skin, it doesn't go into your muscles. Okay, so you have to pack this DNA, this new DNA, in some viruses. It's only vectors. It's not pathogen. There is a capsid, a box with the gene, the new gene in it, and this has a capability to go through the cells into the nucleus to deliver the new gene. And then cells are capable to produce protein that they were not capable to do before. This is gene therapy.
And for hemophilia, it goes to the liver.
Exactly. Right? For the hemophilia, proteins factor are produced by the liver, so we will target the liver cells to produce the factor for coagulations.